Okay, welcome back, class. So today what we are doing, let me check my settings real quick, is we just finished going over different types of reactions, such as endergonic, exergonic, and oxidation versus reduction. So now we're going to talk about the next part of this chapter, ATP and enzymes. And to start, let's talk about ATP. So ATP, or adenosine triphosphate, is known as the energy currency of the cell. And that's the big thing I want you to know about this molecule. ATP is the energy currency of the cell. And while we think about that, I want you to think about the example of if you had to go get groceries. Say you're going to HEB, you go in there, you're grabbing your groceries. In order to actually leave with the groceries, you need to use money to pay for them and to leave with them. So money is our currency. It's the way we get food from, like, say, a grocery store, where we get things. ATP is the energy currency of the cell. So a lot of different reactions, a lot of them proteins depend on ATP for it to be provided energy for that reaction, for that protein or that enzyme to function correctly. And this image here kind of shows what ATP looks like. And one thing I want to focus on is this part right here. These are called phosphate groups. These three phosphate groups, they're all negative. So they all have this negative charge. If you remember from the last unit, we talked about ionic bonds are bonds where a positively charged ion and a negatively charged ion are attracted to each other. So positive and negative like each other. In this molecule, though, ATP, we have three negatively charged um, i- ions that are bound together. These ions do not want to be together. They want to stay away from each other. They want to get away from each other as quick as possible. So this bond here that connects them is what's called a high energy covalent bond. It's a lot of energy in that bond. So ATP is known as the energy currency of the cell because with that bond, when it is broken, it releases a lot of energy. And that energy can be used in endergonic reactions. Remember, endergonic reactions, energy enters the reaction and it gets that energy from sources like ATP, from this very high energy bond. And to show you how that looks, let's look at how ATP releases the stored energy. So removing the endmost phosphate group by hydrolysis, think breakdown. It's really the breakdown by water, by using water. And but for this class, think of hydrolysis as the breaking something. So removing the endmost phosphate group by hydrolysis releases the potential energy stored in the ATP. So if we see in this example, water comes in, is going to break this bond, and when it breaks it, we get a lot of energy being released. We also get what's called ADP. ADP is this molecule that's left over when it has two phosphate groups. So by breaking this high energy covalent bond, we're releasing the potential energy and we get a lot of energy released. Remember, if energy is leaving the system, it's exiting the system, this is an exergonic reaction. Energy is leaving the system, energy is exiting. And this is a very highly exergonic reaction. A lot of energy is being released when that phosphate group is removed. So, how do we get ATP though? So, ATP is produced by cellular respiration, which is a series of chemical reactions that releases energy from sugar, producing ATP from ADP, from spent energy. Think ADP is spent energy. We're going to look at this example here. So we have this food molecule here. With cellular respiration, what it's doing is breaking down that food in a series of reactions. And while it does that, it's able to make ATP. It's able to make that energy currency of the cell. But it takes a lot of control. It takes a series of reactions to make ATP. If you take this food molecule here and break it down in one go, you're going to get a lot less ATP. You can't control the release of energy from the molecule. In the reverse, though, if in photosynthesis, when we're building sugars, we have to use ATP. Since we have small molecules right here, and they are being put together into a bigger molecule that uses ATP, the ATP is spent to build. And again, to go back to exergonic versus endergonic, the top example is exergonic because energy is being released, while the bottom one is endergonic because energy goes in. And we can tell by ATP. So the first one, ATP is going out, and the second one, ATP is going in. So I'm trying to connect all these dots together for you. So in the next slide, if I can get to the next slide, there we go. We're going to look at what's called ATP coupled reactions. So we talked about how ATP is hydrolyzed, it's broken down, and it's released. By doing that, it releases a lot of energy. So ATP breakdown is coupled with endergonic reactions, reactions that need energy. Energy released from ATP breakdown is used to power endergonic reactions. So in the example here, again, we are breaking this bond using water, and a lot of energy is being produced. That energy goes out to other reactions. And we have some examples we're going to see here. So we have one example here where ATP is building something. We have a glucose molecule, ATP is being spent, and we add this phosphate group to the glucose. A lot of times when ATP is being used, it's adding that phosphate group to some other molecule, and it's being what's called activated. So we have activated glucose now that's being added to this short polysaccharide, this short chain of sugars, to make it even longer chain. So ATP can be used to build something. It can also be used to break something too. ATP can be the reverse and then breaking this long polysaccharide to short. So this is an example of how ATP can be used to build a polysaccharide. In this example here, we can see how ATP is used to work on a protein. So this right here, this purple structure right here, is what's called a pump. It's trying to push these molecules right here out of the cell, so or to the top part. In order to do that, it needs ATP. ATP will attach to the protein. ATP would then donate a phosphate group, and that phosphate group would then open up the protein to the other side so these molecules can get out. And then when this protein is released, it goes back to the original state. So there's another example of how ATP is being used. It's being used to help pump molecules in or out of the cell. So ATP can be used for a variety of functions, breaking stuff down, building stuff up, moving things across the membrane, and so forth. But then that's because ATP is the energy currency of the cell. It's how the cell gets energy, it's how the cell uses energy for different reactions. Now, for this question, again, I'll read it off. I want you to pause, think about your answer, and then we'll answer. Which reaction is likely coupled with the hydrolysis of ATP? Pause and think about what the answer is. Okay, so with this problem, if you were thinking about what reaction is paired with the hydrolysis or the breakdown, remember hydrolysis is breakdown of ATP. What I want to do is we know ATP is always paired with an endergonic reaction. So I want to see which ones of these are endergonic. Synthesis of DNA. If you're synthesizing something, you're building it. If you're building something, that means it's endergonic. If you're building, always requires energy. So we know A is endergonic. B, converting proteins into amino acids. So we're going from proteins to amino acids. Remember, amino acids are the monomers of proteins. They're the building blocks of proteins. So we're actually breaking down here. And if we're breaking down, we're exergonic. We're releasing energy. It's easy to break something, and then when you break it, you release the energy that's stored within that molecule. C, joining monosaccharides together. If we're joining something together, we are building. And if we're building, again, that's endergonic. Now we can see here again, ATP is always paired with something that is endergonic. So we know A is right, B is wrong because it's exergonic, and C is right. So that means our answer is E. Now we're going to switch gears. Let's talk about enzymes. Enzymes are also incredibly important within the cell. Enzymes are among the most important of all biological molecules. That's because of two things, or because of really one big thing. Chemical reactions in the cells must occur to sustain life quickly. Without enzymes, many reactions would proceed far too slowly. And again, the big thing here is this top thing. Enzymes speed up biochemical reactions. Enzymes are used to speed up a reaction to make sure it happens quickly, make sure it happens efficiently within the cell or outside of the cell, because some enzymes are put outside of the cell. The enzymes are incredibly important because it helps reactions happen faster. So let's talk about what an enzyme is, though. So an enzyme is a protein that acts as a catalyst. So it is a protein. It acts like a catalyst. A catalyst is something that speeds up a reaction. So an enzyme speeds up a chemical reaction without being consumed. We have the example here where we start with this molecule. Let me show which color is the blue. This molecule here that's going through and being broken up. And we're going to talk about this step by step. So in the first part, we have what's called a substrate. The substrate is the molecule that binds to the enzyme at the active site. And this is where the chemical reaction occurs. So the active site is where the substrate binds, where that initial molecule binds. So this substrate here will bind to the active site, forming this enzyme substrate complex. Once it binds, the reaction can happens a little faster than normal because of the enzyme. Then we get the product molecules are released. The enzyme retains its original form as well, and the reaction products produces products. And these are the products right here, this red and purple block. So substrate is what goes in, product is what comes out. The enzyme does not change though. So the enzyme can go all the way back in the beginning and do the same thing again with another substrate. So all this enzyme is doing is help speeding up this reaction, making it happen faster than it normally would have happened by itself. So with enzymes speed up a variety of reactions. Um, Note that not all enzymes break a single substrate into two products. Some enzymes combine two substrates into one product. And this is an example here where we're taking this glucose molecule, combining it to this chain to get an even longer chain. So enzymes can do a wide variety of functions, and it depends on the specific enzyme. Some will build molecules, some will break molecules down. Some will perform other functions within the cell. It all depends on what that enzyme is. But remember, each enzyme is specific because of its active site. An active site only binds with one substrate. Now, how does an enzyme actually make a reaction happen faster? It works on what's called the activation energy. The activation energy is the energy required for a reaction. So energy required to start a reaction. So even though if reaction is exergonic, sometimes it may need the energy to start. Without the enzyme, activation energy is very high. But when the enzyme binds to the substrate, activation energy is lowered. In this image here, we have the purple line is with an enzyme, the tan line is without. So you can see that in the tan line, we start with our reactants at this energy level and our products way down here. But to get to our products, we have all of this energy right here that we need. So we need all of this energy for that reaction to occur and get to the products. If we look at the purple though, and this again, that energy needed to start the reaction is the activation energy. Now if we look at that purple, which has an enzyme, we have just a bit of activation energy, a lot less than without the enzyme. So that's how enzyme actually functions. It speeds up a reaction by lowering the activation energy, lowering the energy required to start a reaction. And for y'all, that is what I want you to know about an enzyme. It speeds up a reaction by lowering the activation energy from the energy required to start a reaction. Now, cells control the rate of biochemical reactions by limiting the activity of enzymes. In our cells, we don't want a reaction to just continue to happen over and over and over again. That can lead to issues. So our cells have developed ways to stop reactions, stop enzymes. I want to go over those ways. And keep in mind, medicine, a lot of medicine works in this way. It either helps an enzyme, speeds it up, or it stops it somehow. So we have something called inhibitors. Inhibitors lower the activity of enzymes. They do that um, by binding to the molecule and having it stop, like having it unable to function. Binding to the enzyme and having it unable to function. And there's two types of reaction, or excuse me, of inhibitors. Let me get my blue pen now. We have non-competitive inhibitors and competitive inhibitors, and we're going to talk about both right now. So with non-competitive enzyme inhibitors, they change the shape of the active site, the site where the enzyme binds, sorry, where the substrate binds. Cells can produce molecules that binds to the enzymes outside of its active site. That's the key part here. The molecules that bind to the outside of the active site to alter the enzyme shape and prevent the substrate and enzyme from binding together. So if we see in this middle part here, this right here is a non-competitive inhibitor because it's not competing with this active site here. But you can see when that non-competitive inhibitor binds to the enzyme, we go from having this nice square active site where the substrate can bind to the circular active site where the substrate cannot. So we are not competing with the active site. We are simply binding somewhere else on the enzyme to make it where the substrate can't bind anymore. And that's the opposite of what a competitive enzyme inhibitor is. A competitive enzyme inhibitor, binds to the enzyme right at the active site, keeping the substrate and enzyme from binding together. So if it's competitive, it's fighting for that active site. It wants to get in there and it wants to block that substrate. And that's what happens right here. That inhibitor binds to the active site. So then this this substrate can no longer bind. So that is the big thing with inhibitors. They are stopping a reaction from occurring. So in many cases, the inhibitor is the product of a reaction that the enzyme catalyzes. Once there's enough product in the cell, it would be a waste to produce energy, waste of energy to produce more than that product, excuse me. So a lot of times it's the product, whatever the product is, becomes the inhibitor in some way. It tells the enzyme, hey, you made the product, we have the product, we have enough. We don't need any more, we can stop this reaction. So a lot of times the inhibitor is the product. It's going back to tell the enzyme, hey, you can stop, we have enough. Now, enzymes work in very specific conditions. When we talk about temperature, enzymes have what's called an optimal temperature that they work the greatest at. In this graph here, on the y-axis or this vertical axis here, we have reaction rate. Low means slow, high means fast. So we can see in the red is enzymes from the human body. We can see at the very bottom, we have no enzyme reaction. In the very top, at the low temperatures, we have no enzyme activity, and at too high of a temperature, we have no enzyme activity. So we see just at this point, at the very top, we have the most enzyme activity, the fastest rate of reaction. So enzymes have what's called an optimal temperature, which is this temperature right here, where they work the most effectively, the fastest. And each enzyme will have a different optimal temperature. In the next example, we have an enzyme from hot springs bacteria. So you can see their optimum temperature is way higher than ours because they live in a hot, more extreme, hotter environment. So, and the reason that the enzymes a lot of times don't work at high temperatures is they go through what's called denature. They're denatured. Denatured means they're broken down. Denaturing it means that that protein is unraveling, it's breaking apart, and can no longer function. At really high temperatures, proteins denature, they break down. So with enzymes, it makes sense that when you get to too high of a temperature, activity stops because that protein dies. And other chemical factors also have an effect on the enzymes, such as salt concentration and pH. And there's a wide variety of environmental conditions that can have effects on enzymes. These are just some examples. So here's another question for you. According to the graph, at what temperature do you predict the bacteria enzymes become denatured? Look at the graph, think about your answer, and we'll come back. Okay, so we have optimum temperature. We're looking for the optimum temperature. Uh, Excuse me. So we're looking at what temperature do you predict the bacteria enzyme become denatured? So what temperature does the enzyme actually break down? And we know if the enzyme breaks down, it has the lowest activity. And that means it's right here. At too high of a temperature, the enzyme breaks apart and no longer functions. So the answer would be D. And we can tell that because at that point, all enzyme function stops. So now we went over ATP, we went over enzymes. Know what ATP is, know it's the energy currency of the cell, and know it fuels endergonic reactions. We went over enzymes, know that they speed up reactions by lowering activation energy. Know about the two different types of inhibitors. And next time, in the next section, we're going to go over membrane transport.